Let's see. Yeah, where'd you go?
Maybe I should just get down like this.
Present on one knee.
Yeah. Get lower.
Oh, that's like kind of perfect.
Oh, I can do this. Yeah, I can do this.
Should I also get on a knee then?
Yeah, if you want to.
That's so, I must be like so much taller than you. Alright, okay. Yeah, let's do this.
Is it recording?
Yep.
Oh, okay. My name's Ethan,
I'm Cody.
and this is our presentation on the metabolic process of yeast with sucrose and its two monomers.
Yeast, Saccharomyces cerevisiae, is a facultative anaerobe, meaning that it can both cellular respirate and fermentate. Both of these processes produce CO2 as a product, which makes CO2 a great product to measure in the efficiency of its metabolic processes, found by Pepin and Marzocco in 2015. Sucrose is a disaccharide, and its monomers are glucose and fructose. That means that it can be split into sucrose. Sucrose can be split into fructose and glucose. Our hypothesis is based off of this property, as it was hypothesized that sucrose would be the most effective out of the three sugars in metabolic processes and would produce the most CO2 on average, as it has the ability to split and produce more substrate.
We started off by diluting 20 milliliter solutions of each sugar from their higher concentrations all down to a 0.5 molar concentration using water. We then prepared 12 test tubes and split them into 4 groups of 3. So we had a water group, a glucose group, fructose group, and a sucrose group. We put 4.5 milliliters of water into the first group of 3, and then 4.5 milliliters of glucose into the second, 4.5 milliliters of fructose into the second, in the third, and 4.5 of sucrose into the fourth. After all those test tubes had their solutions in, we placed them all into a water bath of 40 degrees Celsius for 10 minutes to incubate. During this 10-minute incubation period, we prepared 12 Durham tubes to use to measure the CO2 that would be produced. After the incubation period finished, we removed all of the test tubes, and one by one, we added 1.5 milliliters of yeast solution into each test tube, and we sealed them with parafilm and mixed them up. After it was mixed, because we did it individually for one test tube at a time, we micropipetted the solution in the test tube into a Durham tube, and then inverted that Durham tube back into the test tube and measured the initial gas space in between the tip and the liquid. After that, we placed the tube into a water bath of 40 degrees Celsius to incubate for 15 minutes. After the 15 minutes was up, we took it out and measured the new volume of gas that was in the tube and subtracted this final from our measured initial to get our final, or total. And that was done for all 12.
Our data was analyzed in Excel, and we had a sample size of 3. These are our results. water yielding the least, fructose the second least, glucose the third least, and sucrose yielding the most.
Our glucose was very close to producing the most. It's kind of hard to tell the difference, but the glucose is just under the 3, and our sucrose is just right above the 3. Our error bars, they are fairly small, which indicates that our results were that our trials were very consistent, and there wasn't much inconsistency with our results.
And any consistency could be due to either initial air bubble measurings being wrong or extra substrate being added by accident.
We can move on here. This was
And a possible reason for fructose being the least is by it having less amount of transporters that are located in yeast, as yeast does have more glucose transporters.
Glucose is more favored as a substrate to use for metabolic processes than fructose is in yeast. Our results from our experiment did support our hypothesis in how sucrose produced the most CO2 and it was the most efficient sugar to be used.
And this could be due to sucrose getting split into both of its monomers and the Saccharomyces cerevisiae being able to use both of those for separate metabolic processes like occurrences.
Our experiment could be improved by increasing our sample size to get a more accurate final result, or it could have been done using, instead of a Durham tube, it could have been a gas syringe, which we would have had our solution in a vial. We would have attached a delivery tube, which was then attached to the gas syringe, and we would seal the vial so that all of the CO2 gas would then float into the gas syringe and would have been very simple to measure.
And this would prevent CO2 being lost in the atmosphere, or just more precise. And so with our results, we can conclude that sucrose would be very efficient in the food industry, as fermentation of yeast is a very valuable product in making bread or brewing alcohol. And Marquise Wesley and their research partners in 2016, and Gord Yuke and Van Oudenarden in 2009, and Greg and Travinoso in 2004 found that sucrose splitting was very beneficial, as its splitting and the uptake of the monomers that were produced costed no extra in the metabolic process. And comparing our experiment to another very similar experiment done by Pepin and Marzocco in 2015, Pepin and Marzocco had very similar results to us.
They tested the rate that yeast would use sucrose as a substrate to undergo fermentation, and they compared that with how it would do with glucose and then how it would do with fructose. And despite hypothesizing that sucrose would be the worst due to it having to be hydrolyzed by the enzyme invertase to be then used in glycolysis, they found that it had no effect on the rate of the metabolic rate it was put through, and it still came out on top.
Indeed. Here are articles cited. Thank you very much for your time.
Thank you.
Oh.